Hello, good afternoon.
Good afternoon.
Afternoon. Okay, I think we can start the session now. Okay. Good afternoon to everyone who have joined with us today. So, today is our fifth meeting, I believe, in our third creative camp for urban agriculture. And today we will have the Mr. Jason and Miss Regalia from
Regale.
Rigel from Muntinlupa National High School, who will give us a lecture on the aquaculture and also the algae culture. Okay, so without further ado, please, Mr. and Mr., you can deliver your session. Thank you.
Start now. Good afternoon, everyone. We are from Muntinlupa National High School, Philippines, and we will be presenting to you aquaculture and algaculture. What does it mean when we say aquaculture? It is the controlled production of organisms that normally live in water for a better yield and economic viability. There are different types of aquaculture and there are different practices that we do here in our school when it comes to aquaculture, and later on we will be sharing them to you.
So aquaculture, when we deal with aquaculture, it is producing fish in a limited and controlled, for example, in ponds, tanks, aquariums, lakes, and others. It is a human intervention to increase aquatic productivity through cultivation activities and other processes. It also multiplies the population and growth of biomass and improve the quality of aquatic biota products to obtain profit. So
Yes, the key there is uh, producing or culturing fish in limited and controlled environments. The benefits of aquaculture. First is food production, an increase in food production. It's one of the sustainable development goals of UN. Stock enhancement, fish for recreation, ornamental fish production, and industry material production. Here are the factors to consider for good aquaculture practices.
first is the site location. Second is pond preparation. Next is fish stocking, feeding management, and water quality.
That's the model that we have. And for our, that's number one, have shown a while ago, we will considerations. First, you need to focus on the water resources, and it's typically surface water or groundwater to supply aquaculture operations. So you need to look for a site that will really have a good supply of water for your operation. And secondly, the source of water must be free of contamination from fertilizers, manure, pesticides, or other chemicals. And of course, there must be a testing of the water quality. Here are some examples of aquaculture sites.
So it can be for ponds. You have example, the basin.
And varying sizes. As you can see, these are all artificial ponds or man-made ponds.
So these are examples of the the location in the lakes.
Okay, so we have ponds, lakes, and we also have tanks. Tanks can also be varying in sizes and appearance. It could be installed at home, like the one we see here, the aquarium, or it could be outdoors. Another example of a tank is a bioreactor. The one shown on the upper left portion is the photobioreactor that we have made as a tank for our algaculture. Another consideration for fish stocking. How do you choose the fish?
So choose the fish with the same average size. Next is it should be physically good, healthy and lively, have a good growth performance. performance. And the time for stocking should be early in the morning or late in the afternoon and should be adapted. Fish stocking, you need to check the quality of the fish before it is introduced into the pond or on the site location. So it should have a certificate of production based from the bureau that covers your country. Here in the Philippines, we have the Bureau of Animal Industry and the Aquatic Resources. And those fish should be free from diseases and should undergo stress tests before it is released in the pond.
This material is obtained from the presentation of Uniati 2018 by Biotrop and Sea Creative Camp first batch. And we also want to share that we need to take note of these formulas. To find the total biomass within the culture system, we need to use this formula, number of fish times average weight, to find the stocking density. So you have the formula total biomass in kilograms all over or divided by pond area times the volume. And to find the number of fish needed to place in culture rearing unit when you are given the initial stocking density, you have to use this formula. First, you need to multiply the volume or surface area of rearing unit by the initial stocking density in kilogram per square meters. And there you get the average weight of the fish in kilograms.
To find the number of fish required when given the final stocking density in kilograms over meter per second or cubic meter and the survival rate known, the formula would be final stocking density times volume of the culture system over the average weight at harvest times the percentage of survival.
Okay, so that's the stocking density in kilogram per square meters or cubic meters. To find the volume in cubic meters, you need to multiply the length, the width, and the depth, and that will be in meters. And to find the final number of fish when given final stocking density, harvest weight, and rearing unit volume, you need to use the formula final stocking density times volume of the rearing unit divided by the average weight of fish at harvest, and that is in kilograms.
And to find the initial number of fish when given the percentage survival and the final number of fish, the formula is final number of fish over the percentage of survival.
There you have it. We have the formulas. Next slide. Next slide is about the feeding management. You can see a table wherein we have the first column as the number of days up to harvest, and the second column, the feed amount in percent from biomass. So for 3 to 15 days, we have 5%. 15 to 25 days, we have 4% feed amount. And for 25 to 45 days, 3% feed amount. And 45 to 60 days, 2.5%. And up to harvest, that's 60 to up to the harvest, we have 1.5% from biomass.
With a protein use of 28 to 30%, and you need also to consider that you need to buy feed from the reputable feed supplier, and also the fish feed can be utilized within about 3 months only to help maintain the feed quality and safety because it is already exposed to some factors in the environment.
Yes, that's why you need to consider buying feeds that have longer shelf life as compared to other feeds. So you need to buy from trusted or reputable feed suppliers.
Also, you can have your own computation for the feed stock that you can provide based on the need of the type of fish that you will be releasing into your site location.
And another table, as shown here, is the water quality. We have the parameters used, and those are the dissolved oxygen, maybe as measured by BOD meter, and the pH parameter, the ammonia
Content.
ammonia content, the temperature, the alkalinity, and the hardness of the water. And we have here on the second column the optimum concentration, which should be greater than 4.0 milligrams per liter. And another one is 6.5 to 8.5 pH, and that is in between. That could be, yes, and ammonia is 0.15.
0.15 milligrams
It
per
should be
liter.
less than 0.15 milligrams per liter.
And the temperature should be 27 to 30 degrees Celsius and should be measured daily. For the alkalinity, you have the minimum of 0.5 milligrams per liter to 100 to 400 milligrams per liter. And it is measured several times a year. For the hardness of the water, you have minimum of 0.550 milligrams per liter to 100 to 400 milligrams per liter and also measured several times daily. So there are technologies nowadays that applies automation of the testing for water quality, and that could be applied also in the aquaculture and algaculture. So this is to automate the monitoring of the dissolved oxygen, pH, ammonia, and temperature, and to assure the maximum yield at the end of the production time.
And we must also consider adding probiotics. Why do we need to add probiotics? It's the good bacteria that will enhance the quality of the water, and that is also to avoid the presence of the bad bacteria because of the exclusive competition among those organisms. So what probiotics may be used? I think actinomycetes are also used to enhance the quality or to promote the good flora in that tank. And yes, you have to consider also the required frequency of monitoring. And that's for water quality. Now we move on to the next slide, which is the culture. So that's also a part of aquaculture. Algaculture, from the word alga, which means the alga or the algae. Aquaculture is a form of aquaculture involving the farming of species of algae. And we know that majority of algae that are intentionally cultivated fall into this category. And we have what we call the phytoplankton, microphytes, or planktonic algae. Actually, they're the oldest species or the oldest organisms on Earth. So that's already a local wisdom that we use. Why do we need to culture algae? What are the benefits? First,
It can produce various products such as food, such as the gelatin that we eat. For animal feeds, it is one of the prime component for feeds that are used for animals. Soil fertilizers, medicine, major component of the production of medicine, cosmetics, and health supplements. So it can be used as feedstock for biodiesel, which can be converted into biofuel also, which can be used for vehicles with a very high biofuel yield. And it can be used to purify water.
another benefit is that it really absorbs greenhouse gases and reduces carbon dioxide as it releases oxygen. It's really helpful, especially in climate change, in mitigating the effects of climate change, as it absorbs the carbon emissions and it also, yes, the greenhouse gases as well. And it also has a great flux tolerance and photosynthetic efficiency with limited nutrient requirements and flexible cultivation conditions. So it's really resilient despite the fluctuation in temperature, in any circumstance where you grow the algae. It will be resilient despite the changes in the atmosphere or in the environment. That's why it's good to cultivate algae because it requires less maintenance. That means if you cultivate algae, that will equate to green algae harvest, and green algae harvest equals generated income.
Algae cultivation can be done in a variety of environments, including first, open ponds can be categorized into natural waters like lakes, lagoons, and ponds, and artificial ponds or containers.
Yes, they can actually grow even in open and natural environments. But it's also good to cultivate the algae in closed ponds, and that's why we came up with a photobioreactor. It's a cultivation system that actually costs more than the open pond, but of course, it's more efficient and considerably less than photobioreactors for similar areas of operation. So under the closed pond it could be a photobioreactor, which is an equipment that provides a controlled environment for the algae. And we can see that high productivity is expected.
For the open pond system, one of the most natural methods to culture algae is a free environment, which has a sunny areas would be preferred to get maximum production since the sunlight is needed uh, in the production of algae, which is a requirement in the photosynthesis. Considering their low maintenance and construction cost, open ponds are the best option for conducting initial experiment on a large scale. So an open system are categorized into natural, which are small ponds, lakes, and lagoons, and artificial ponds, which can be done in a container and tanks.
For the examples of open ponds, we have the raceway pond. It could be artificial or natural pond. So this raceway pond is an artificial pond that is used in the cultivation of algae. There's also another pond uh, which uses a paddle wheel to make the water flow continuously around the circuit. So here in the raceway pond, we can see that there are rectangular grids, and each rectangle contains one channel in the shape of an oval, like an automotive raceway circuit. That's why it's called a raceway pond. Another example is the circular pond. Circular, which has the shape of a circle, as we have in Japan and in China. On the left side, we can also see that there is paddle wheel to make the water flow continuously as the one shown here on the bottom. And here in China, they have paddle wheel also. And in Japan, a circular pond.
Seemingly like agitator. That
Yes, and the motion is really needed to be able to make the algae grow better and to avoid the stocking up of the algae in the system. What do you consider when you design an open system? The first one is you should design an open system with mixing patterns. Why is it important that there must be mixing? It plays an important role in the distribution of sunlight because, of course, in the growth of the algae, sunlight is really needed. And it's, of course, the essential nutrients needed by the algae will be evenly distributed.
Next is the mixing technology. Mixing can be achieved using different technologies. For example, you have the gas sparging and the magnetic beads have been used in algae culture. So there are different types of technologies that you can use. These are the already available ones, and you can also design your own depending upon the need or the type of container that you will be having.
In our case, we used a sparger run by motors, and the motors are run by solar panels. And you must also consider the depth of open ponds, and the typical depth ranges from 5 to 100 cm. Or another type is the closed system, or another term says closed loop system, which is not exposed to open air. And that's good because it avoids the problem of contamination by other organisms blown in by the air.
So the widespread production of algae by using this method for biofuel, since there would be a direct connection between the production or from the closed system going to the harvesting part and in the production of the biodiesel. It is highly controlled production since it is controlled system, it would be easy for the farmer to monitor the different of the algae. It also allows integration facility with other facilities like combined heat and power system. And most popular method of closed loop system is the photobio reactors.
In this picture, I think the algae shown here is the green algae, which is the we use Chlorella vulgaris, and we will show them to you later on.
Okay, for the next part, example of a closed system is a photobioreactor. There are different types of photobioreactor. The tubular photobioreactor, the Christmas tree photobioreactor, the plate photobioreactor, horizontal photobioreactor, and foil photobioreactor.
So many types and your own type of photobioreactor. But these are all the physical parameters to be considered in creating your own photobioreactor. First is the temperature. An intermediate value of 18.20 degrees Celsius must be employed.
So this 18 to 20 degrees Celsius can exceed, but not too much. It may range to until 28 degrees Celsius. But the most preferable temperature should be 18 to 20 degrees Celsius.
And lighting must also be considered. Artificial lighting by fluorescent bulbs is often employed for culture maintenance and experimental purposes. But you may also use LED to make it more efficient, like the ones that we used in our project. And we use blue light so that we can have greater frequency, which means greater energy to be captured by the algae.
And the mixing, it should be noted that in the oceans, cells seldom experience turbulence, and hence mixing should be gentle. So the mixing tool that you need to use should be gentle enough to facilitate the production. The following methods may also be used: bubbling with air, plankton wheels or roller tables, or about 11 p.m., and gentle manual swirling. So you can also devise your own depending upon the type of container that you will use.
Yes, that's why the mixing must be consistent and it should not be too violent to avoid dying, so that the planktons or the algae will not die because of mixing. I'll bring to you the best practices of Muntinlupa National High School, and we will be showing you some of the aquaculture projects here in our school.
First project we have is aquaponics, a large-scale aquaponics and a small-scale aquaponics.
The large-scale aquaponics uses PVC pipes, and here man-made pond, as you can see in the picture. And the small-scale aquaponics, which was done for the purpose of research, made out of used PET bottles or polyethylene gallons, and they're growing tilapia fingerlings and gotocola or Centella asiatica as plants. There's scale aquaponics in our school, and fish pond. This is the algae bioreactor that we have. Now we are on the process of automating the harvest of the algae. We have different models of photobioreactors already. The one that is solar powered, and this one in the picture is using alternating current. The other one is DC or direct current. Here you can see that blue lights, blue LED bulbs are used, light emitting diodes, to facilitate the growth of the algae. Any more questions? Thank you so much for listening.
Thank you.
Okay, thank you very much, Mr. and Miss, for your explanation. So just for your information, the Algaculture project is the one that makes the Muntinlupa the first prize. Yeah, we get the first prize for the first SEA Creative Camp on urban agriculture. Okay, so does anyone have a question?
We are open to answer any question that you have.
Yeah, you can also ask the question in bahasa and we will try to translate it to English. Oke, dari SMA Tenggarong.
Tenggarang.
Tenggarang.
SMA 1 Tenggarang.
Oke, silakan. SMA 1 Tenggarang.
Our question is what kind of green algae that you cultivate in your school?
It's the Chlorella vulgaris, the species Chlorella vulgaris. There is another algae, which is the blue-green algae, which is the spirulina, but we have not yet tried that. Right now, we are focusing on the Chlorella vulgaris, or the green algae. It's the one that is used in medicine for height, for increasing the height of the sun. And it has so many benefits as what we have presented to you a while ago, and that's Chlorella vulgaris. Thank you.
Okay.
Okay, thank you very much.
Thank you.
Okay, thank you. Okay, so any more questions? Or I believe while we are waiting for some questions, I believe SMK 5 Jember would like to also share the practices on soil-based gardening, right? Is it correct, SMK 5 Jember? Halo, SMK 5 Jember?
Kalau kamu rame, Gina mana bisa dengar?
Halo, SMK 5 Jember.
Ya.
Di ba may tanong doon na feeding management?
Oh, pero sino ba ito? Diba last time? Naunahan ko yun.
Ano? Sila
Okay, we have questions. What is the difference between aquaponic and hydroponic?
Yes, the difference between aquaponics and hydroponics is the one that we have mentioned a while ago, the container, and as well as the medium of planting. For aquaponics, it is a part of aquaculture, while hydroponics is not. Aquaculture for aquaponics because it uses the fish or culturing fish as well as cultivating vegetables on top. So there is a cycle that happens. That's why there must be a submersible pump to catch the water, and the water with the wastes obtained from the fish will be transferred or will be used to water the plant, which is grown on a growing medium like pebbles. So it's a soil-less planting as well as hydroponics. Both of them are soil-less planting processes, but the difference is the growing medium. For hydroponics, the growing medium is different. So you must have a solution containing all the nutrients needed. So the main difference exactly is the nutrients. Like for hydroponics, the nutrients is already prepared. The solution is the one that supplies the nutrients for the plant. And the growing medium is also different. Unlike for aquaponics, The growing medium can just be rocks or pebbles, and the nutrients will come from the waste of the fish, the waste material from the fish. So both of them in aquaponics, the plant and the fish, both of them benefit from each other. So that's it. The main difference, the container, the growing medium, the components. So in aquaponics, the components are the fish and the plant, while for hydroponics, it's just the plant being cultivated. So those are the differences between hydroponics and aquaponics.
Okay, thank you. I hope it answers your questions. SD. Yeah. Okay, perhaps any more questions from the audience?
Excuse me, sir.
Yeah.
I want to ask from SMA Sadun Garang.
Okay, sure.
How
Please.
many harvesting chlorella burgeries in your school, and what relation return between cultivate algae with aquaponic? Okay, thank you. And how much
You can harvest it.
okay, how much you can harvesting the chlorella?
Okay, for the first question, how many
Harvesting chlorella burgeries in a year?
in a year? Okay.
Yes.
It's 5 gallons per, that's
Photobioreactor.
per photobioreactor. And so we have the data here that, okay, I will look for the data. Yung difference muna ng, the difference for the difference between the aquaponics and the algaculture, to explain to you first, habang hinahanap yung data. Difference ng aquaponics and algaculture.
The difference between aquaponics and
Algaculture and photobioreactor.
algaculture and photobioreactor. So for the algaculture and photobioreactor, it is a controlled system that you can monitor the different physical parameters. So we are also producing that in a closed photobioreactor, which we get the needs of the algae from the LED lights. So the difference between the aquaponics and the algaculture would be in the aquaponics you can use other types of organism, but for the algaculture itself, for the photobioreactor utilizing photobioreactor, it will only be exclusive for green algae.
And for the algae biomass concentration, we have 0.25 kilograms per cubic meter. And if you compute that, that is 400 metric tons of cultured algae
per year.
So that's the amount that if you're asking. But we can sell 1 liter of algae for up to 3,000 pesos per liter.
Which can be converted
How much is that in Indonesian money?
So Indonesia.
So in Philippine peso, it's 3,000.
How much dollar is it?
How much dollars?
That is
$3,000. 3,000.
That is approximately $60 per liter. So it is somewhat expensive. That if you produce a liter of that in a weekly basis, you can really earn money. And utilizing the photobioreactor, you can assure that the production would be on a maximum rate, since you will be harvesting 2 to 3 times a day of the desired volume of your photobioreactor.
And if you will be using it for biofuel, that will be around 5,000 gallons of biofuel per acre per year. And there are so many benefits why we culture algae, so that's it. It could be from the biomass or from the oil or from the alcohol that can be obtained from it. So it's depending upon your purpose why you want to culture the algae. Any other question that we did not answer? We might miss another question.
My next question is there relation between algae cultivate and hydroponic?
Algaculture and hydroponics.
Algaculture connection
And hydroponic.
and hydroponics. Both of them are culturing or cultivating a living organism, but the difference is that in hydroponics you are cultivating a plant, while for the algaculture, instead of plant, it's a plant-like organism. And both of them, the similarity is that both of them contribute to the emission of oxygen, and that is really helpful in the absorption of greenhouse gases as well, because the benefits of algae are also the benefits obtained from other plants grown in hydroponics.
In our school, the purpose why we focus on the green algae, which is the Chlorella vulgaris, is the fact that here in our area, which is the tropical areas, seldom that we encounter countries or organizations that focus on algae production. So that's why we wanted to promote algae culture, not just here in the Philippines, but also in other countries, because of the great benefit that we can have from that. It has a close relationship to other types of hydroponics or algae culture, but using algae would give us great benefit that at present times is not yet utilized.
And both of them, hydroponics and
can be accommodated by a photobioreactor, since the consideration is the size of that algae, because it is a floating algae. Unlike with the spirulina and the Chlorella vulgaris, they are microscopic. So this spirogyra is a bigger type of algae. That's why you need to consider the design of your photobioreactor.
Yes. Yes, sir. Thank you. Thank you, sir.
Thank you.
Okay, thank you SMAN 1 Tenggarang. Okay, so perhaps any more questions? Or SMK 5 Jember, are you ready to present your presentation?
Kailangan pala naka-online pa rin tayo kahit nawala na tayo.
Halo, SMK 5 Jember?
Ya,
Kailangan tutukan natin.
Oke. Jadi presentasi hari ini? Halo?
halo, yes.
Yeah. Will you give your presentation today?
Okay.
Okay.
We will be closing our...
Okay, so I believe SMK 5 Jember is ready. So Muntinlupa, can you give your closing remarks?
So, we would like to thank everyone, especially the Simeo Barotrog, for the opportunity. And we in the Philippines are very glad to be part of the third batch of the Sea Creative Camp on Urban Agriculture.
Rest assured that we will be willing to help all of you. So if you have any questions, you may contact us. Thank you, Mr. Harritz, for considering us once again to be able to share to all of you the things that we know about aquaculture and algaculture.
Okay, we from Muntinlupa National High School, thank you and good afternoon.
Okay, good afternoon. Thank you very much. Okay, so that's it for Muntinlupa. I hope it inspires you with new ideas, and we are really hoping to see some new algae culture from Indonesia. Okay, now we move on to SMKN 5 Jember. Okay, we have passed the presenter to you, so you can share your desktop.
Yes. It's done.
Okay, thank you. We've got your presentation. So you can begin now.
Excuse me. Can you see my PowerPoint?
Yes, we can see it clearly. You can just play the PowerPoint. Play the slideshow, I mean.
Yes. Yang ngomong. Iya.
Mhm.
Ini dia masuk. Dia masuk.
Yeah, we can hear you clearly already. Just
Yes.
please. Oops.
Nggak bisa.
Oke, udah dengar kok, Bu. Tinggal ini aja, F5 aja biar full screen gitu presentasinya.
PowerPoint-nya sudah terlihat?
Sudah terlihat, cuma apa di full screen kan aja F5.
Ya. Oke. This my student will be presented.
Ya, terima kasih, Bu. Silakan Ibu bisa dimulai.
Excuse me.
Ya.
Yeah. Excuse me. Good afternoon. Thank you for the second chance that Mr. Harith give us. Let me introduce our team.
Sorry to interrupt.
Firman and Amin. We are students of State Vocational High School 5 Jember in East Java, Indonesia. We will share our experience about vegetables and fruits production in State Vocational High School 5 Jember. Kind of vegetables product including sawi, lettuce, cauliflower, spinach, kangkung, bean leaves or lembayung in Javanese, leek, onion, paria, cucumber, sweet corn, and baby corn. Kinds of fruit including strawberry, orange, banana, and melon.
Slide how to produce vegetables. How to produce vegetables. Polyculture method. Intercropping. We apply this method because the field in our place is limited, while we learn to produce various kinds of vegetables. Soil minimum tillage. The type of soil in our place is hard in the dry season and sticky in the rainy season. So we apply this technique. We do extensive soil treatment at the beginning of the rainy season, which is plowing and making beds. The next planting season, we only loosen the soil on beds. Organic and chemical fertilizer. We combine of organic and chemical fertilizer. We use more organic fertilizer to improve soil structure, while we add chemical fertilizer according to the stage if needed. Return weeds and harvest waste to the soil. We return the weeds that have been cleaned and the rest of the harvest on the beds. If the conditions are still wet, we place them on the surface of the bed, which also function as a mulch. We do this in dry season to maintain soil moisture. Non-chemical pesticides and refugia plants. We are committed to producing healthy fresh vegetables, so we don't use chemical pesticides. We apply biological pesticides, biological agents, and planting refugia plants for pest control. Harvest continuously every day. We schedule the production of vegetable crops until the harvest is done every day. morning and harvest time, direct selling by students. Sales are carried out directly to customers. Which is done by students in the morning. Fruit plant import, tabulam pot for orange and strawberry, some else in the field, orange, banana, melon. The fruit plant have not been routinely produced, so the harvest is also not routine. Our fruit marketing is done directly according to customer demand.
There is the expression we can share at this Southeast Asia Paris Game Forum. Thank you. Kamu terlalu cepat. Presentasi itu slow.
Okay, thank you very much for your presentation.
Maybe any question?
Yes, any question for SMK 5 Jember? And after this, please send your presentation to me. I will give you my email address, and don't forget to put your names in there because we are planning to give the certificates for the presenters.
Excuse me, my name is Sultanul Islam Nur Fitrah Tunggoh from Senior High School 1 Tenggarang. I want to say in bahasa.
Ya, silakan, Mas.
Kami kan siswa SMA.
Ya.
Dan tidak mengerti apa namanya intercropping. Bisa dijelaskan apa itu intercropping?
Okay, thank you. Intercropping itu intercropping adalah sistem pola tanam polikultur atau tumpang sari kalau di bahasa Indonesia. Jadi dalam satu lahan atau dalam satu lahan itu tidak hanya satu jenis tanaman tapi berbagai jenis tanaman. Bisa dalam satu bedeng itu terdiri dari 2 atau 3 jenis tanaman dengan tipe pertumbuhan yang berbeda. Mungkin itu bisa diterima. Enggak kedengeran.
Ya, terima kasih. Pertanyaan selanjutnya. Ya.
Mantap.
Metode yang dipakai apa saja? Apa metodenya itu konvensional? Atau ada hidroponik? Atau aquaponik juga? Atau verticulture?
Karena ini soil based production, artinya berproduksi berbasis tanah, kami menggunakan metode konvensional semi modern. Karena untuk yang semi modern itu menggunakan media tidak murni tanah, tapi bisa media yang lain seperti arang sekam, pupuk dan media yang lain. Kemudian dengan organik itu bisa dikatakan sistem konvensional yang modern. Karena untuk pertanian yang awal itu organik murni karena sistemnya berpindah lahan. Itu tanpa menggunakan pupuk, tanpa menggunakan pestisida. Sementara setelah proses budidaya itu penggunaan pupuk dan pestisida kimia itu ditambah. Bisa dikatakan untuk yang modern itu back to nature, kembali ke alam dengan pupuk dan pestisida kimia, dengan mengoptimalkan kearifan lokal termasuk agensi hayati yang bisa dikatakan dalam ekosistem adalah musuh alami. Bisa itu prosesnya seperti alih simbiosis, parasit, kemudian predator, kami menggunakan itu melalui penanaman tanaman Ya, jadi kalau di slide yang sebelumnya itu di antara tanaman semangka itu ada tanaman bunga. Tanaman bunga itu dimaksudkan untuk menarik agensia hayati supaya nantinya hama itu tidak langsung pada tanaman budidaya melainkan pada tanaman bunga. Warna merah, kuning, ataupun oranye itu menarik beberapa jenis serangga yang berbeda. Mungkin itu. Jadi kami hidroponik ada, tetapi karena ini yang kami share itu yang materi sebelumnya yang minggu lalu, yaitu vegetables and fruit soil based production. Jadi kami men-share pengalaman kami yang menggunakan media tanah langsung di lahan, bahkan untuk yang kedepannya kami tidak semua di lahan terbuka, tapi ada di greenhouse gitu. Walaupun di greenhouse kita juga tetap ada media tanah, media sekam, arak sekam, dan juga media kokopit. Kokopit itu serbuk sabut kelapa yang dimanfaatkan sebagai media tanam. Jadi kalau diperhatikan di slide, nah ini slide ketiga ini ada tanaman bunga. Itu fungsinya untuk menarik serangga, menarik musuh alami, menarik agensi hayati supaya menjadi predator, antibiosis, parasit dari hama. Ada yang bisa saya sampaikan lagi?
Oke, dari kami terima kasih.
Thank you.
Oke, mungkin ada pertanyaan dari yang lain?
Can we ask a question?
Please go ahead.
My question is, how can you ensure a sustainable crop management without the use of chemical fertilizers and pesticides?
Okay, thank you. Do you get the question, SMK 5 Jember?
Excuse me, can you repeat once again your question? Sustainable about what? I'm sorry, I will try to explain in bahasa. Jadi untuk pertanian berkelanjutan itu kami sudah memulai pertanian berkelanjutan dengan meminimalkan penggunaan bahan kimia, baik itu dari pupuknya atau chemical fertilizer. Kami menggunakan sedikit dari dosis jika diperlukan saja. Sementara untuk yang pupuk, untuk yang pestisida, kami menuju ke zero chemical pesticide. Jadi menuju ke minimal sekali penggunaan pestisida kimia, mengingat untuk sustainable kami berkomitmen untuk menghasilkan atau memproduksi sayur dan buah segar yang sehat. Sayur dan buah segar yang sehat itu otomatis residu pestisida di dalamnya itu sedikit, sehingga kami memang meminimalisir untuk penggunaan itu. Walaupun di sekolah kami juga memproduksi benih yang itu intens menggunakan pestisida kimia, tetapi di dalam greenhouse, sehingga diharapkan residunya tidak terlalu banyak untuk tanaman di sekelilingnya. Jadi untuk sustainable.
Can I try to translate from what you said first before you continue? Okay, so basically what SMK 5 Jember said is that they are trying to minimize the use of the chemical fertilizer, and also for the pesticide, they are trying to not use the chemical pesticide at all. Although they have some nurseries that use the chemical fertilizer, but it's it in greenhouse. Is it something like that, SMK 5 Jember?
Yes,
Okay.
I think it's enough.
Yeah, please continue with your explanation.
Uh, saya kira itu yang untuk pertanyaan berkelanjutan karena kami juga masih belajar. Jadi kami masih pada taraf yang sangat rendah, yaitu taraf meminimalkan penggunaan bahan kimia baik itu pupuk dan pestisida. Nah, untuk yang mengontrol atau untuk pengendalian hama penyakit, kami menggunakan penanaman tanaman refugia. Jadi tanaman ini kalau dulu mungkin distilahkan sebagai tanaman barier, yang itu sebagai perangkap hama, sekaligus juga tempat hidupnya predator, agensia hayati, atau bahkan juga parasitoid yang fungsinya nanti akan menghambat pertumbuhan atau perkembangan dari hama. Jadi beberapa siswa kami sudah mengamati bahwa memang dengan penanaman refugia itu di situ ada predator. Jadi ada beberapa lebah, ada beberapa kumbang itu yang memakan hama. Jadi ada yang kemudian menjadi predator pada beberapa fase. Ada yang predatornya pada fase nimfa, ada yang pada fase lava, bahkan kalau itu parasitoid maka dia akan menginfeksi sehingga hamanya akan mati. Jadi paling tidak memanfaatkan lingkungan sekitar. Kemudian kalau untuk pestisida biologi atau pestisida nabati, kami memanfaatkan tanaman yang ada, terutama tanaman mimba, karena di mana di Jember ini tanaman mimba sangat banyak dan itu sangat bagus karena kandungan azadirachtinnya.
Tanaman apa?
Dan juga untuk yang jadi ada 3 itu, kami menggunakan pestisida biologi, jadi pestisida nabati, ramuan dari tanaman yang bersifat racun untuk hama, tetapi aman ketika kemudian hasil produksi tanaman produksi dari sayur dan buahnya itu aman dikonsumsi. Kemudian memanfaatkan agensia hayati dengan penanaman refugia itu.
Okay, I will try to explain in English, but because I'm not a biology person actually, so please correct me if I'm wrong. Okay, so in order to try to prevent the use of chemical pesticide, the SMK 5 Jember have tried several methods to prevent the insects or pests to eat their diseases. The first one is the biological pesticide, which is the use of apa tadi Bu? Pestisida biologinya Bu?
Mimba, Azadirachta indica.
Yeah, that's it. Okay, and then also they are using the nabati pesticide, which is the use of limbah tadi Bu ya, waste plants, and also to use the planting of refugia that was aimed to attract bees and bees that eat the pest gitu Bu ya.
Iya, untuk tempat hidupnya predator, parasitoid, atau musuh alami dari hama.
So to attract the natural enemy of the pest, something like that. Okay, I hope you get what I mean. Mungkin lupa.
Thank you.
Okay, thank you.
That satisfies the question.
Okay, so any more questions from the participants? ada pertanyaan lagi, bisa langsung di unmute atau bisa chat aja. Silakan. Ibu, kalau tanaman buahnya di SMK 5 Jember apa aja, Ibu?
Tanaman itu bunga kertas atau zinnia, kemudian marigold, bunga matahari.
Buah, punten.
Oh, buah.
Fruits ya.
Buahnya stroberi kami masih mencoba. Kemudian jeruk itu tabulampot dan sebagian di lahan, pisang Cavendish, kemudian melon. Melon ada yang memang untuk buah, ada yang itu hasil samping dari produksi benih. Jadi benihnya diambil, diekstrak, kemudian buah melonnya dipotong-potong dijual dalam bentuk potongan. Seperti itu.
Okay.
Buah segar yang sudah slicing, artinya yang sudah dipotong-potong.
Oke, makasih.
Dan dikemas.
Mungkin kemarin sih ada program ini Smart Bee dari Biotrop untuk sekolah buah. Nah, sebenarnya di Malang itu ada, di Bali Jestro itu ada berbagai macam ini Bu, apa varietas jeruk sebenarnya.
Iya,
Mungkin
kami sedang ongoing di Smart Bee. Jadi yang di Smart Bee itu kami mencoba di jeruk, anggur, pisang Cavendish, stroberi walaupun di dataran rendah, kemudian sayuran, untuk sayuran termasuk mungkin yang akan kami masukkan lembayung karena ternyata demand-nya banyak, permintaannya banyak.
untuk apa Bu, lembayung?
Lembayung, ya.
Oh.
Daun kacang yang masih muda. Jadi kalau biasanya di Jawa sini, di Jawa ya, maksudnya di Jawa Timur, lembayung itu biasanya hasil pangkasan dari kacang panjang. Cuman kita karena baby, jadi dalam bentuk baby lembayung. Daun kacang panjang.
Oke, siap. Makasih Bu.
Kembali.
Oke.
Permisi,
Silakan.
saya ingin tanya lagi.
Silakan.
Ya.
Apa dengan pestisida?
Ya.
Pestisidanya bebas.
Terus apa ada kombinasi dengan pestisida kimia? Kalau ada, perbandingannya berapa persen?
Kami mulai membuat pestisida itu zero. Jadi kita tidak menggunakan pestisida sama sekali. Jadi kami menggunakan pestisida dari mimba itu tadi. Jadi mimba, dan ada beberapa ramuan lain, mulai dari bawang putih, kemudian biji sirsat, itu kita mulai menggunakan. Jadi kita tidak menggunakan pestisida kimia. Kalau penggabungan kami ya tadi itu, jadi pestisida nabati, pestisida kemudian menggunakan agensia hayati, dan agensia hayati itu kami pelihara melalui tanaman refugia. Tanaman refugianya adalah tanaman bunga kertas, bunga matahari, bunga marigold, kemudian ada juga kami yang akan mulai itu bunga knob atau bunga kancing. Kemudian kami juga akan tambahkan selasih karena itu merupakan tanaman yang bisa menarik lalat buah karena kami sudah mulai untuk memproduksi buah. Nah, kalau yang lembayung tadi yang di slide yang ini kami panennya ini lembayung. Jadi di umur 10 hari kami sudah panen.
Baby longkin.
Jadi mungkin itu. Jadi kami mulai, kalau untuk pestisida kimia sendiri hanya untuk produksi benih yang kami gunakan di beberapa greenhouse. Jadi harapannya supaya pestisida itu tidak terlalu residunya tidak melebar keluar. Kami menggunakan sesuai anjuran. Karena anjurannya berapa, kita juga harus lihat atau konsentrasi dosis dan lain sebagainya. Kita harus lihat dulu dari gejalanya, apakah masih bisa ditoleransi sehingga dengan dosis yang rendah, ataukah sudah harus dimusnahkan karena kalau dosisnya terlalu tinggi takutnya hamanya akan semakin resisten terhadap pestisida. Demikian.
Oke, bagaimana SMA Tenggarang?
Pestisida nabati buatan SMK 5 itu apa bisa digunakan untuk semua tanaman? Pestisida nabati buatan SMK 5 itu apakah bisa digunakan untuk semua tanaman? Atau hanya tanaman tertentu? Terima kasih.
Yang mana?
Bisa saya jawab, Pak Arif? Pestisidanya ini karena yang sudah pernah kami coba itu kami menggunakan limbah. Limbah itu mempunyai kemampuan yang cukup luas untuk membunuh atau untuk meracun ya, karena pestisida itu kan pada hakikatnya racun bukan obat. Jadi untuk meracun serangga itu cukup ampuh, terutama untuk yang ular. Jadi kalau untuk yang limbah hampir luas jangkauannya. Jadi spektrumnya cukup luas. Tapi ada beberapa seperti jamur, misalnya Beveria bassiana, itu harus melalui proses pembiakan dulu. Setelah melalui proses pembiakan, kemudian diambil, diencerkan, disemprotkan dan ke serangga, bahkan serangga dengan kutikula yang cukup tebal pun bisa terinfeksi jamur ini, jamur Beveria. Jadi jamur Beveria ini hanya inangnya atau berparasit pada serangga, bukan pada tanaman.
Baik, terima kasih Ibu. Bisa minta tolong di-share gimana caranya, cara membuatnya.
Cara pembuatan yang limbah atau yang Beveria?
Yang limbah. Limbah, Bu, limbah.
Untuk pestisida limbah itu sebenarnya cukup mudah. Jadi ambil saja daun limbah, kemudian diekstrak, diambil airnya, lalu kemudian didiamkan 1 hari supaya mengendap, diambil yang bening, kemudian diencerkan sesuai dengan konsentrasi. Jadi tidak hanya limbah sebenarnya, ada tembakau. Tembakau itu kalau sudah di dalam kondisi kering, sudah dilajang, itu bisa digunakan juga sebagai pestisida. Jadi bisa direndam, kemudian dalam 3 sampai 5 hari, kemudian airnya diambil, lalu diencerkan, bisa juga untuk pestisida. Begitu juga yang ampuh itu biji sirsak. Jadi para pembuatannya itu biasanya melalui proses fermentasi atau proses pembiaran dulu selama minimal biasanya 24 jam diendapkan untuk diambil sarinya. Jadi biar racunnya itu keluar dulu dalam air, kemudian diambil bukan yang endapannya tapi yang murninya airnya itu diambil, kemudian diencerkan baru disemprotkan ke tanaman.
Takarannya berapa banyak, Bu? Seperti limbahnya berapa banyak, airnya berapa liter?
Kalau dosis kita tergantung serangan ya, minimal itu pembuatan biasanya kalau limbah itu biasanya perbandingannya 100 gram dan 1 liter. Kemudian akan diencerkan. Jadi biasanya harusnya di blender biasa. Jadi di blender dengan air daunnya 100 gram, airnya 10 liter. Kemudian disaring, didiamkan selama 3 sampai 5 hari, tergantung kita tingkat racunnya sampai sejauh mana. Bisa hanya 24 jam, itu juga bisa langsung digunakan. Memang kelemahannya kalau untuk pestisida nabati itu aromanya kurang sedap. Jadi ada kelemahannya di situ.
Bu, terima kasih atas jawabannya.
Sama-sama.
Same.
Terima kasih Bu jawabannya. Kemudian ada pertanyaan
Ya,
lainnya?
terima kasih Pak Ari.
Oke, kalau tidak ada pertanyaan lainnya, saya kira mungkin bisa ditutup, Ibu.
Oh iya. Sudah ini Pak Ari?
Oh, oke.
Baik, kami akan tutup. Terima kasih atas kesempatannya.
Oke, terima kasih Bu. Nanti mohon PPT-nya dikirimkan ya Bu ya. Sama dikasih nama-nama siswanya yang presentasi.
Iya.
Oke, makasih Bu.
Terima kasih Pak Ari.
Okay, so I think that's it for today's everyone. So once again, thank you to everyone who have attended this session, and especially to Miss Regairo from Muntinlupa and also Mr. Jason and from SMK 5 Jember who have already shared with us the best practices of aquaculture and also the soil-based vegetable gardening. Okay, so with that said, I think we can end this session today, and hopefully we can meet again for the next session at Thursday at 18 October to talk about the oyster mushroom cultivation. So once again, thank you and see you again at the next meeting. Wassalamualaikum warahmatullahi wabarakatuh.